a deal for you. All right. I do what some of the other professors I do. If you 70%, at least 70% of you fill out the OCQs, I will give you one free question in the final. That's good, you know.
Yeah, she said she's skipping it. I'm really worried about the exam. I just feel that really well. I got so many points on that. You took off like all the wrong. So you're like, that's just a big thing. I'm like, yeah. So I'm like, oh, because it's small. So the angle, number, and everything. Took off points for all of them. Like, that exam, walking out. So what's the deal? Stop doing a mistake. I was so mad. Could you tell me to chill? Because I don't want your everything to do today. Or what? Stop telling me that. Whatever. I'm gonna start. He goes like, what's a concentration? To draw like questions. Like there's like this main problem, and then this main problem that I'm working through. Maybe. I mean, they're not like bad. I mean, some of them I was like, really? Oh my god, I was so mad. I did the whole quadratic equation, and then I realized for this one, I put 1 minus 2x. I did everything for it, and it was 1 plus. And I really wrote 1 plus 2x here. And I was like, no, I just put fix. And then just wrote down the I'm not doing the whole quadratic. No, I'm not doing it again. It's too much. I don't remember the order that we were studying. I was trying to remember, like, you remember when you were talking about it? Because that's really close. I think it was like really bad. I think it was also I We could just finish all of them today. I do. Or even all of these books. That's insane. Are they gonna like sell that?
You guys finished? You guys finished? Thank you.
I tried it some of mine. I got to 3.
Oh, I'm sorry, but there isn't,
See, I just can't get it.
you're out of batteries. This? Let's see.
It's just, I love her.
Okay, all right. Can you guys hear me? Is it good? All right, I am going to try and finish the course today. All right, so I might be going a tad fast, but it's okay. So that you don't have to come on Thursday. Okay, is that good? Yeah, so I'm gonna try and do it. I know I can do it because I did it for the other class. And they were all very happy, so. All right, we talked about fertilization. The sperm meets the egg, you form a zygote. All right? And then the zygote that's formed right here will then travel, up the oviduct, and then it will implant in the uterus. So development, that means development of the egg, mitosis of the egg, is happening the moment after fertilization and while it is traveling along the oviduct. It can happen here or here because, as you know, the gonads come in pairs. All right. Now, so I'm going to show you what happens. Okay, so the first part, after fertilization, the egg divides into 2. That is mitosis. All right, and it looks like this. Right? Okay. So this is the zygote. This is the sperm fertilized egg, and it undergoes mitosis. So one division will give 2 cells. 2 divisions between 4 cells and then 8 cells. And then at about 96 hours, you find that you form what is called a morula. A morula is a solid ball of cells, okay? A solid ball of cells. That means there is no space between the cells, all right? And then you find that that becomes the blastocyst. Now the blastocyst is different from the morula because by the time you reach the blastocyst, you find that the cells have migrated. It's no longer a solid ball of cells without space between the cells. It is now a hollow ball. It means that a lot of the cells have moved towards the periphery. So this is the hollow ball here, and this is a solid here, all right? The hollow ball is called the blastocyst. Now, if you cut open the blastocyst, you will see this hollow in the middle. There's a layer of cells outside. But then there is this mass of cells here. This is in purple, all right? And let's look at it more closely. Yeah, it looks like this. It's in purple. Now, in the early days, scientists didn't know what it was. They just thought, well, this is a random clump of cells there, all right? But then they found later on that it was more important. This mass of cells actually gives rise to the fetus. But in this particular portion, at this time in history, they just called it the inner cell mass, not the fetus, although it becomes the fetus, all right? So you have the first stages here, the early stages, that is part of cleavage, which is mitosis. Remember, not meiosis. This is mitosis. Once fertilization has occurred, you find there's mitosis, all right? So this is a cleavage, and then that is next followed by gastrulation, right? So here is your blastocyst here. You will find if you're looking at the microscope, a lot of these cells begin to move. They really literally move cell by cell to various parts of the embryo. And you get what is a process called gastrulation. All right. And if you look at the cells in gastrulation, you find that there are 3 distinct layers of cells. They're distinct because they are aligned, so that one layer is called the ectoderm, the middle layer is called the mesoderm, and the innermost layer is called the endoderm. Each of these will give rise to different tissues, okay? So this is gastrulation, and here, this is the most beautiful photograph I've ever seen of implantation. This is about 6 to 14 days, all right, so after fertilization. So you can see this is a mass of cells, and this purple thing here, that is the uterus. Now, I mentioned to you earlier on that this mass of cells, all right, more than 100 cells, come from one cell. This cell. This is the egg, and it's fed with nutrients and mitochondria from a whole series, a whole corona of nerve cells. This one cell will divide and divide and divide to give you what I just showed you. That is the cells for implantation. So from one cell, you get these cells, over 100 cells, right? And all this time without input of energy from any other source. So all the energy that's required for making this clump of cells actually came from one cell. That's the most remarkable thing, all right? Now this here comes, this now takes the place of the corpus luteum. Remember, the corpus luteum is formed after ovulation. So now for this clump of cells to attach to the wall, right, you need the hormone progesterone. If progesterone levels drop, you will find that this very young embryo will fall. Right? Now this is a very interesting history. All right? Prior to, say, 20 years ago, if you wanted an abortion, you had to go for various types of abortion. Many of them are surgical. They have their side effects. They're also expensive. All right? Now this drug called RU486 was a drug that was discovered in Europe. And it was a very effective drug. All you have to do is take it, and 3 days later, this embryo will drop off. That is a spontaneous abortion, okay? And it was, in Europe, it cost only $15 for 30 tabs. It was very inexpensive. But it was not available in this country. You could not get it in the United States for a very simple reason. The Europeans who synthesized this did not have, they did not seek a patent for it. So there was no money to be made. If there's no money to be made, nobody wanted it. So it was prevented by the pharmaceutical companies to import it to the United States. All right? However, good old President Clinton, on his last day in office, as you know, whenever you are president, you want to push something through that is against the agenda of your supporters, you do it on the last day. So on the last day of his presidency, he permitted RU-486 to be imported to this country. So now you can buy it over the counter. This is called a morning after pill, okay? Which is quite remarkable because this doesn't cost hardly anything, all right? And it's safe, okay? All right, next we come to this process that I will tell you about again. This is called apoptosis, where cells die at a particular time. Now, so let's go on here. I just mentioned to you that there were 3 layers of cells: the ectoderm, the outermost cell, the mesoderm, and the endoderm. Now, they are destined to become different tissues. So the green area here, the ectoderm, becomes the skin. They differentiate into skin, the nervous system, and the retina. And then the middle layer, the mesoderm, differentiate into muscles, connective tissue, kidney, blood vessels, reproductive organs, and then over here, the endoderm, they become internal organs like the lungs, the liver, and so on. So these cells, because of their locations, all right, they become different cell types, okay? And this is sort of important because it used to be thought that, well, cells become different types of tissues based on their genes, but after this we find that it's not so. It's based on their locations. So in other words, if you have an egg there, or an early one like say this, and you stick a needle in there and then you stir it all up, and then you change the orientation of the cells, guess what? You will get dead tissue or you will get just crazy tissue. All right? Their location within this blastocyst determines what kind of cells they become. All right, then next you find that the scientists look to see what we can learn about early embryogenesis. It's difficult to learn about early embryogenesis in humans because how do you find 1 egg or 2 eggs in the human system? You can't, all right? It's very difficult. So they look to frogs. Remember I told you this is human development, but we refer to some simpler organisms like frogs, like sea urchins and so on. So if you look at a frog embryo, you can see there are 3 layers. The green layer, this is the ectoderm, and then the middle layer, and then the innermost layer. And what they found was when they look at vertebrates, like chickens, like birds and so on, there's always this thing here, which is like a chasm on top of the embryo. And it's very consistent. They called it the primitive streak. In other words, it's found in all vertebrates. All right? And basically what it is that you see this layer of cells, all right? And as it grows, it invaginates, and then it forms a solid tube that runs across the length of the embryo. So it looks like this. All right? If you're looking down, bird's eye view, you can see here is the embryo, and here this is the primitive streak. Initially it's short, and then it extends all the way across, and then it begins to fold, and the tube is called a notochord. When it folds up, it becomes the neurotube. So this little thing here, the primitive streak here, actually defines the length of the embryo. Right? So it will begin to seal up The anterior part will develop into the face, the larynx and pharynx, the pharyngeal arches here, which remind us of our fish, shall we say, ancestry. And then this area here will seal up. It becomes sealed, and it looks like a tail at one point. All right? If it doesn't seal and it branches out like a fish, then we have a disease called spina bifida, which is quite common. Now, so if you look further at the embryo, you will notice something else, that there, in addition to the embryo, you can see the embryo here in chicken and the embryo here in humans. In addition to the embryo, you see 4 extra embryonic membranes, all right? And they are this blue area here called the amnion, all right? Colloquially called the water sac, all right? And somebody says their water just broke, that's what it's referred to. You see this water sac here. It's not water actually, it consists of nutrients, consists of waste, but people refer to it as water sac. Right? And then there's a yolk sac, you know what that is, right? The first site of red blood cell formation, and then you find that there is this green thing here called allantois. Together, these they form the umbilical cord blood vessels. All right? So this is what they look like in humans. This is what they look like in chicks. And then the outermost layer in chicks is called the chorion, the shell. In humans, it is the placenta. Right? So there are 4 extra embryonic membranes: the amnion, the yolk sac, the allantois, and the chorion. This is in chick, and this is in humans. Right? Now, when I had a lab with this class made way back when, and we could see fertilized chicken eggs, and we can see all these parts. It's really, really, really interesting, very beautiful. Okay. Now here is another view. Here is reptile, bird, chicken, and here is mammal. You can see the striking resemblance. Right? In mammals, the amniotic sac is a bit larger in this particular phase, but you can see the umbilical cord and reduced here, expanded here. All right. So when you look at all these embryonic membranes, then you come up with the reason for them being there. They originate from the embryo, but they're not part of it. They assist in the development of the embryo. They provide protection. They transport nutrients and waste. And then ultimately, they're discarded. Right? So the 4 embryonic membranes, they do have functions, as you can see here: protection, nutrients, waste, and so on. And if you look further, you will come up with the conclusion that most scientists came up with, and is a dissimilarity in the development of the embryo. It's reminiscent of the stages of evolutionary history. In other words, they came up with this phrase: ontogeny recaptures phylogeny. In other words, the development of the embryo from fertilization to gestation goes through all the adult stages of the evolutionary history of the phylogeny. Okay? So if you look at this again, right, when this develops, it goes through many of the stages that you see in the development of humans. It does not mean that we came from chickens. Okay, very important there. Just to show you more embryos and how strikingly similar they are. If you discard all the embryonic membranes, all right, you will see this in fish, this reptiles, this bird, this mammals. In other words, they're very, very, very similar. Even seasoned scientists have difficulty telling these apart without the extraordinary embryonic membrane. Now here's another view, it's very similar. This is fish. That fish looks like this, but the embryonic fish looks like this. All right. Here's salamander, here's tortoise, here's chicken, here's rabbit, and here's man. So in other words, at this particular stage of development, we look very, very much like all the other lower organisms. Now here you see where we are. We have talked about gamete formation, eggs and sperms, gametogenesis. fertilization, cleavage to form the solid ball cells, gastrulation to form the hollow ball cells, and then we begin to form organs. And then when these organs differentiate, we have different organs, we have tissue specialization and so on. So we've covered all that very quickly. Right. Next we go to something that is a political hot potato. All right. And usually when we have elections, this becomes really a hot topic. And what this is the stem cell saga. All right. So this is how it works. All right. So you know that there are stem cells. In other words, there are cells that can differentiate into more complicated cells, but they all start at the beginning, where they are embryonic. All right. That means they haven't been given the signals to differentiate. Right? So, for example, you have adult stem cells. These are the cells in the bone marrow. You can stimulate them under the right conditions to become other kinds of adult cells, like blood cells, white blood cells, red blood cells, and so on. Right? And then this is the most controversial, embryonic stem cells. In other words, these come from aborted fetuses. But to harvest them so that they be alive for transplantation, you have to take live aborted fetuses. And this has caused a lot of controversy because when you see the films and all that, it looks really quite disturbing. Right? But the ultimate goal for embryonic cells and all other types of stem cells is that you should be able to, or could, or wish for, that they would differentiate into cells that you want. For example, if you have Alzheimer's or Parkinson's, or you're, crippled by some dreadful nervous disease, you can get those embryonic cells to differentiate into nerve cells. Okay. Now, that is wishful thinking right now. There's a little bit of progress, but not much. And then there's another approach, and that one looks much better. That is to get your stem cells from the umbilical cord. All right. So this is something you guys might think about in your next phase of your life. All right. So the idea here is, should we use stem cells? And if so, what kind of stem cells? All right. And how do we convert stem cells to the type of cells we want? Suppose, for example, we want blood cells. We want blood cells because at this moment in time, we are always buying blood cells. We can't manufacture them. Right? Now in Singapore, they claim that they have been able to differentiate embryonic stem cells to blood cells, but it's on a limited basis. It's not like they have reached a stage where they can sell it, and they don't have a blood bank. All right, that comes from stem cells. Embryonic cells, this is very controversial. People have taken aborted fetuses, different tissues from aborted fetuses, and implanted them. All right? For example, in Europe, but not here, they have been able to take aborted stem cells, planted them next to brains of patients who are demented, and some of those cells have grown, they have multiplied. That's step one, to get those cells to grow. All right? Hopefully, one of these days, they could cure Alzheimer's or Parkinson's or something else. Right? But the journey from embryonic stem cells to a differentiated cell is slow. Okay? So, and then here, now these actually work quite well. Embryonic cells, and you take them and you put them in the vicinity of the heart, not somewhere else, but where the heart is. They will differentiate a little bit into heart cells. Their location is very important. There's something about the location that allows them to differentiate into a nerve cell or a heart cell. Now, so what's the problem? The problem is most people don't like the idea of taking embryonic stem cells. You can see the fetuses, all right, and you're crushing them, all right. So that's a little bit disturbing. The one that seems to be working quite well is cord cells. When you have children in the next so many decades of your life, you can, after delivery, ask your doctor to freeze your cord cells, all right? Because those can differentiate somewhat. That costs money, so you have to arrange it before time. They will freeze it for you, minus 90 degrees, all right, and it will last for a while. All right. So there are always problems with stem cells. Adult stem cells are working better now because they're able to use some of these adult stem cells and transfer them to patients with leukemia. All right, you can zap all your cells and then introduce some stem cells from another person. It's worked in a limited way. The reason I wanted to introduce that is because people talk about stem cells and they all have opinions, but they don't really know all the different types of stem cells. You know, maybe you don't want stem cells from aborted fetuses, but you might consider stem cells from bone marrow, from a friend, a relative, or you might consider stem cells from your umbilical cord. So those are different. Now here, you're looking at something that is very much more common today, and that is in vitro fertilization. At one time, it was hit and miss. It was very expensive. It is still very expensive, but it's not hit and miss so much. All right? So here is your fertilized egg, one cell, all right? And then you can see division, all that, until blastocyst. So this is what you want to implant into the woman, all right? It's at this level of about 100 cells, not younger, not older, so you can get this thing to grow and become a viable fetus. This is pretty tricky, all right, and very expensive, but the success rate now is a lot higher, all right? And you will find that many in vitro fertilization patients will have triplets, quadruplets, or even more. And the reason is they are very expensive procedures. And so you want to implant not one or two, but usually about 6 or 10. And then you slowly abort a couple of them, okay? So that is another way of getting pregnant. Now, then beyond that, we are just growing. This is a human embryo, it's 4 weeks old. You can see this is a spinal cord, it's beautiful. At one time we looked like that. We not very attractive, but we do have something that looks like a tail. It pulls up as we grow, so we end up without a tail. This is the foot pad. This is the head, the pharyngeal pouches. This is a very lovely photograph. Now, this is 4 weeks old. You can see the head here. Remember, the brain is the first organ to develop. So therefore, the take-home lesson is you should always be healthy. A lot of people will say, oh, well, I mean, I'm not 5 months yet. I don't know where that magical 5 months came from. Maybe it's because it's visible. You're visibly pregnant at 5 months. But the brain forms very early, all right, at the very beginning. So you should make sure that you're not drinking a lot of stuff. All right, so pharyngeal pouches here, developing around. Here's your neural tube. Right. This is where you want to make sure that the tube is sealed. If it's not, if it's like a fishtail, you have a disease called spina bifida. And that is not good because it causes all kinds of urinary problems and reproductive problems. Here's 5 weeks, okay? Here you can see the brains forming, gill clefts. Now here I want you to see, this is the limb pad. This is the upper limb pad, okay? This is the tail here. Now here's a really important point. So we talked about apoptosis before, and that is programmed cell death. In other words, it's not necrosis. Necrosis is due to a lack of oxygen or something like that. This is apoptosis, where it's programmed cell death. In other words, those cells are programmed to die, and the purpose is for sculpting. It's just like here, all right, you have a tadpole with a tail here, all right? And you know, those of you who have ever seen tadpoles, you know, they have a tail 1 minute, and the next minute, overnight, tail's gone, all right? And they become a four-legged frog. And that's because these cells here die by apoptosis. They just very quietly, no inflammation, they die. Right? And this is the way the body uses to sculpt organs. So most organs start as lump, you know, tissues. without any particular shape. But if you kill certain cells at certain locations, you will get good shapes, just like this here. All right. So the organ is, this is your limb pad, all right, and it looks like this, continuous. And then these cells here in purple, they die. And now you have separated fingers and toes. Okay? And this is a process that's used repeatedly, but using it, looking at it from the point of view of the formation of individual digits is really quite dramatic. You can see this through the ultrasound. Right? So initially it's continuous, right? And then those cells in between die by apoptosis. Now you have separated fingers and toes. Okay? And this is the way all organs are shaped. So that's why it's called sculpting. Now here you can see 5 to 6 weeks old, right? More development, the head, you can see the growth of the head, in other words, the brain exceeds the growth of other regions. Now, when you are 5 to 6 weeks old, the features, all right, you reach a stage where you suffer from nausea, all right? And it's colloquially called morning sickness. In other words, you're nauseous, you vomit, and you generally don't feel very well. Right? Now, it's called morning sickness, which makes you think it happens only in the mornings, but it can happen any time of day. Okay? So you just feel, unwell, so to speak. You can't stand up straight without feeling giddy. All right? So lots of women tend to look for solutions. They go to see their doctors, and the doctors would give them something for nausea, which makes sense. All right? Except in this one instance, it was not good. All right? You find that the drug that was used in the 1960s was a drug called thalidomide. Okay? And it was meant for something else. It was a sedative, a calmer. All right. It was used to treat anxiety, insomnia, gastritis, and tension. Right? So they thought, well, if we can treat all that, why don't we use it to treat morning sickness? Except that was really a bad choice. All right. Many infants, over here it says 5,000 to 7,000 were born with phocomelia. That's an underestimate because this was used worldwide, except the United States. The simple reason was in the United States at that time, you needed to have the drug tested for 10 years. The minimum period was 10 years. So we didn't have that. However, it affected the wives of our troops, because we had troops in Europe, especially in Germany, all right? And the wives of those troops, they had, kids with phocomelia, where on the exterior you could see they didn't have limbs, but on the interior there were many, many deficiencies. Okay, so the thalidomide problem was really very, very pronounced in Germany. You can see that some of the survivors, they had legs, but short arms or no arms. So, the manifestation of arms and legs and toes and fingers depended on how much thalidomide was taken and for what period of time. So if they were taken later, they would probably have legs. If they were taken earlier, they may not. So there is a rather sad story. Here's a girl. Many parents found that if they were missing one set of limbs, the other set, like in this case, the legs would work very, very well. This poor kid didn't have arms. Okay. Now, so this was banned. Thalidomide was banned, and everybody heaved a sigh of relief. But then it came back to haunt us. And it was very interesting. So about 10 years ago, somebody proposed the following, and that is, well, we have cancer. This is a whole different area altogether, not pregnancy. This is cancer. As you know, cancer, huge lumps of cells, and they're growing very fast, which means that they're fed by plenty of blood vessels. Right? lots of blood vessels. You can tell if you look at a mass of tissue that has been excised, you find that it's very bloody. All right, and then doctors know, surgeons know, they're very familiar with this, that you can remove that, and then you think you've removed every bit of tissue. However, they come back. You have cancer again in 5 years, all right, or less. So they said, well, this is tough. Why don't we do something else? Why don't we shrink this? That mass of tissue, maybe 5 inches across, we'll shrink it to 2 inches across. And then if we excise it, then we have a greater chance of removing cancerous tissue. Right? How did they shrink it? They added thalidomide. And thalidomide will shrink the blood vessels. Right? So therefore, these will not grow as well. They will become much smaller. And then now you can get rid of this lump. And then you can celebrate and say, hooray, you got rid of this lump of tissue. All right? And then they found something new. They found when that happened, all right, the women who had that procedure, who had cancer removed, they had thalidomide babies. Why? Because the thalidomide stayed behind. Doctors told them, well, you know, it's soluble, it will be gone in 6 months. Well, they were not gone in 6 months, so therefore they stayed behind, and those mothers had thalidomide babies. Right? So that was not a good idea. They also used it for treating TB, tuberculosis, and they forgot, or they said patients didn't follow instructions. So lots and lots of babies in South America were born without limbs. So the take-home lesson from that is, you know, sometimes even though doctors have very good intentions, they're great, you know, at saving lives and so on, it doesn't mean that they know everything. They don't. We all find that there are limitations. So the best thing to do is not to take a lot of stuff during your pregnancy, all right? It's just very heart-rending. I'll tell you a story, all right? I used to take my daughter swimming. I was like every other mother. I would sit there by the side of the pool, and all these kids would be swimming, and I would pretend to look interested, and I would read my books and all that. And so I took my daughter swimming. She loved to swim, just like a fish. You know? But there was this boy who was really like a fish. He was like a dolphin, you know, this not a splash. Now the swimmers, if they are good, will not go like, you know, a big splash in the water everywhere kind of thing. They glide through the water. Beautiful. So my daughter was swimming with this boy, and I was just like, oh, he is so good. Wish my daughter could swim as well. She was a little clumsier, little splashes here and there. But anyway, one day I was looking up, and I saw this boy come out of the water, and I thought, that's something strange. He's very short. It turns out he was a thalidomide baby. He had no limbs. So he had no arms and no legs. So when he came out of the water, he was just waddling like a penguin. And that kid was just remarkable. He wasn't embarrassed. He wasn't, you know, shy. He was just, his mom was raised him really, really well. Because he just stood there, put on his, you know, armament, so to speak, his hands and his legs. And then he was about 6 foot taller, you know, very well raised. But I asked his mom, I said, what happened to him? And she said, oh, good old thalidomide. Right? And that was it, thalidomide. Right? So be careful. Now then another drug came along just to remind us that there are still more drugs out there that could be harmful. And that is something called diethylstilbestrol. This was made in Indianapolis, made by Lilly, all right? Now they're a good firm, I'm not saying they're a bad firm, it's just that they're close to home. And this drug was used as a synthetic estrogen, so it was administered for people who had difficulty getting pregnant because they had low levels of estrogen. What happened with this was this caused this clear cell adenocarcinoma. Now, this was a very difficult case. This case went on for like 30 years. Right? What it was that this drug affected the ovules of the person who took it. So you take this drug, all right, and you're fine. but not your grandchild. So this was a very difficult case to prove. Everybody said, no, how can we say, this is related to this? This is like 40 years apart, okay? But it was a nasty case because it affected the drugs. So what I'm saying is that be very careful what you take. It might affect your child directly or a few generations past. Now here is something else. This is fetal alcohol syndrome. Now most people will say, oh, well, I will stop taking cannabis, so something very seriously bad. But maybe just a little sip of alcohol will be okay. And this is what you might find. This is fetal alcohol syndrome. Look at the results. This is from drinking a lot of alcohol over a period of pregnancy. You have all this poor coordination, problems with vision, poor judgment, intellectual and learning disabilities, heart and kidney problems, mood swings, deformed limbs or fingers. All of these are not very good. I mean, there's a certain phenotype. If you look at their eyes, they have the same epicanthic form as people with Down syndrome, but they are not a product of Down syndrome. They are not trisomic for 21. Okay, so this is something to keep in mind, that you don't want to designate that something is not so bad after all, and then get into a situation where you produce a child that's totally not well equipped for life. I will tell you a story, a real story, okay, about this woman. She gave birth to a retarded child, mentally challenged, as they would say, and she was very interesting. Most people would say, oh, I shouldn't have done that. I shouldn't have drunk a quart of whiskey a day, which is what she did. She drank a lot of whiskey every day. And then she had a mentally challenged child. And her excuse was, I didn't know. Well, nobody told me I couldn't drink, a quart of whiskey a day. And then, this is was really interesting. She sued the alcohol distiller, and everybody said, don't waste your money. Everybody knows excessive alcohol is bad for you. You're wasting your money. You really are wasting your money, so you should stop that nonsense. Right? And guess what? She won. Why? Because if you look at a bottle of alcohol, there are no warnings. Honestly, go to the alcohol shop and you can see nice wine, red wine, white wine, rosé, whatever. There is no warning. It's not like cigarettes, where it says, oh, be careful, you might get lung cancer. Right? Nothing. And she won the case. I thought, oh, good for her. I mean, not good for the disability, but good for her. A challenge, I guess. All right. So here is alcohol fetal syndrome. And now here I want to point out to you, this is fetal alcohol spectrum disorder. Now this was found by a woman scientist, and she was studying the effect of alcohol on children development. And she had all these photographs of these children pinned to her board. And then she noticed something. She said, this is very interesting. Most of the kids with that phenotype, this is a phenotype, right? Like you see in this fetal alcohol syndrome and in trisomics. She said, it's very interesting. All these kids are Indian, American Indians. All right. So what is the common factor amongst these? All right. And she found, look at the number, 33% higher than in the rest of the population. And what happened was she dug deeper and she found that all these children came from poverty. Their parents have no money, they live in very poor conditions, they don't have food, no education, and most of these kids, they just exist. All right? And most of the parents, they exist by drinking alcohol in large amounts. Okay? And that was the common denominator. Poverty bred a high incidence of alcohol consumption, and therefore they suffered from this fetal alcohol syndrome. Right? So sometimes when you look at people who are in the lower level in terms of the economic spectrum, you have to remember it will affect many aspects of their life. All right, next we talk about the sex chromosomes. As you know, in humans, the most important determinant of sex is this region of the Y, so-called SRY, the sex determining region of the Y chromosome. Okay? Now, if you look at the tubes in vivo, I colored them so that the pink tubes, they are the mesonephric ducts. No, they are the paramesonephric ducts. And in the blue tubes, they are the mesonephric ducts. Now, so when we are there at early stages of about 5 weeks, you find that we have the ability to be either sex. Right? We can be male or we can be female, depending on whether we have the Y chromosome or not. If we have the Y chromosome, we will have testosterone, we will have this region, the SRY region, and then you'll find that this blue region, this is the mesonephric duct, they will develop into normal male organs. Right? However, if we have no SRY, but we have DAX1 instead, then it is the pink ones here that develop into the female reproductive organs like the uterus and the vagina. So that is interior here, and this is the exterior. All right. So this is what we look like at 7 weeks. At 10 weeks, we look the same. We can't tell, we can't distinguish between male and female. But then as birth approaches, you can see the development of the penis. Now, then we come to a critical point. There have been lots of debates, controversies, fights, people constantly questioning when does life begin. Because it's important to know when life begins. You know, like if you kill something at age 1 month, is that real life? Or is that not real life? Is that a person? Is that not a person? So this went on for years and years and years and years. And then now they have finally decided, as of now, when a fetus is 8 weeks old, that is when all its organs are there. They may not all be well developed, but they are present at 8 weeks. That is officially dubbed a fetus. Right? So that is the beginning of life. Okay? And everybody has to adhere to that, whether it's political or pharmaceutical or whatever. Okay? And you can see at 8 weeks they have eyes, nose, mouth, a big brain, genitals are formed, okay? And so on. So remember, 8 weeks. Okay? Now, so these are various photos to show you as the fetus grows. They get bigger. So this is all mitosis. The organs just grow larger by mitosis. Now then here, what you see is this branch thing here, those are the fetal blood vessels, and is surrounded by maternal blood. Right? And what this shows you is that the fetal blood vessels are very close to the maternal blood, but they do not mix. Right? However, some small molecules like immunoglobulin G, they would travel to the fetus from mom. Right? So if mom is healthy, she would give her IgGs to the fetus. Right? And small things like microbes, like for example, think about rubella, German measles, HIV, they can cross the placenta. But remember, the large cells do not cross. Right? It's only small particles that will cross. Now here is week 10. Right? It looks kind of big, right? Now here you have the fetus has eyelashes, eyebrows, some have hair, some have nipples, some have fingernails. Now here is the important thing. Bone replaces cartilage. Okay? So in other words, the fetus hardens. Okay? And this determines the kind of abortion you can have. Before the formation of bone, you can sonicate a fetus, and it will be fine. But once bone forms, if you sonicate that, then the bone fragments will spew out like little spears, and it will damage your uterus. So you can no longer do a non-surgical abortion. Okay. Now this is very interesting because this is the point where it will frown, like what they call the pop eye look. You know, they kind of frown at you, and then they open one eye and close the other. It's very charming. All right. So 12 weeks. Okay. Now here I want to point out the fact that there's always this political debate, you know, who should and who shouldn't, and when and how and why people should have abortions. What criteria should we use to determine it? One was the age. So they determined, well, okay, we have 8 weeks, that's the fetus. After that, you have to be careful, and it depends on the state. So there are state rules, there are county rules, there are federal rules, and all that. So they say, well, maybe another criterion is if the mother could look at the ultrasound. I mean, just go to the doctor's office, look at the ultrasound, and say, I'm fine with killing that fetus, or I'm not fine with killing the fetus. All right? So you know, you look at a picture like this, how can you tell? I mean, you know it's alive, you hear its heartbeat, but you know, it's not that simple. And now there are new technology. Now this is 2D, all right? This is 4D. Look at that. This you can see, right? It looks just like the baby that you get afterwards. So they thought, oh my, if you look at this face, can you abort it? All right? And then once they saw this, they said, oh, we'll change the law altogether. We don't want to see such a clear picture. So they abandoned that idea. All right, so this goes on much larger, 14 weeks. This is 20 weeks. Okay, this is just a comparison between 16 weeks and a little older over here, 38 weeks. So you can see they're very, very similar, just this one here at 38 weeks, which is full term. This is larger. Now here is when we have to talk about abortions. As you know, there are different states with different rules. Generally speaking, 24 weeks, that's the end. It can't be older than 24 weeks. This is in the United States. So late-term abortions, 21 to 24 weeks with the stop at 24 weeks. Now, if you look at the numbers, okay, if you remove a fetus at 24 weeks, there's a chance, 35% chance of survival. If you wait one more week, there's a 50 to 70% chance of survival. If you wait another week, you have a 90% chance of survival. So you should not be aborting at 26 weeks. That, you know, it has a 90% chance of survival. Okay. Now you can see here, this is 20 weeks. Man, it's kind of, you know, human. It's big. It's cramped in here. Then you find that the fetus gets a little hairy, very soft hair. That's called vernix caseosa. Now here is 28 weeks. You should not be aborting at 28 weeks. This is able to survive outside the womb. This is referred to as the age of viability. Now some countries, not the United States, they allow abortions up to and beyond 28 weeks. But if you look at this, this is life. Okay. Now this is when this is 2 weeks before delivery, and you can see the fetus will grow as large as the uterus. It doesn't go beyond that, but it's really getting crowded here. So at birth, the typical American baby is about 20 inches long and about 7.5 pounds. That used to be the norm. Now they're getting a lot bigger. Now here you can see the orientation of the fetus. Remember, we start out like this, where the head is further away from the cervix. But as the fetus matures, you see the head turns around. So that at birth, the head comes up first, like a squid. So let me show you the different stages of birth. The three. This is the first one where the head is here, pressed against the cervix. All right. Now you want to notice what you can't see. The blue line, which is gone, that is the amniotic fluid. Most of the amniotic fluid is gone by this time, but the bag is still there. All right. And the doctor will break the bag, or you will break the bag spontaneously. That's when people refer to breaking of the water. Somebody will say, "Oh, my water is just broken, goodbye," and off they go to the hospital, okay? And then here is the mucus plug. It's not mucus, it's like your endometrium is bloody, all right? And that comes off. And here is blocking the cervix and the canal that goes in here. All right, so when this comes off, it comes off like about 2-3 tablespoons of blood, then you know it's time to go to the hospital. Okay? So dilation of the cervix, the dilation begins, contraction, and usually what happens is that you get to about 10 centimeters, and that is a good size for the head to be able to pop out. Right? So this is stage 1, dilation of the cervix, and then stage 2 is the expulsion, what do you call it, expulsion of the infant. In other words, it would turn around so that it sees the floor. So the first view of life is the floor. Okay? I don't know why they don't make it more interesting, like paint some Mickey Mouse or something on the floor, but they don't. There it is. And then the third stage is the expulsion of the placenta. So sometimes the placenta here, this comes out after the fetus and is referred to as this, that's the placenta, the afterbirth. So this is the afterbirth. All right, you can see the placenta detaches from the uterine wall. All right, and here is the umbilical cord. Now this is what you want to preserve if you want embryonic cells for later life. All right. When you're 40 years old and you have cancer, maybe this is what you would depend on. Okay. So here we are. Wrinkly face. We are not very attractive when we are first born. We are slimy, we cover with hair and blood. So we're not very attractive here, but we get more attractive with age. Okay. All right, and then now after birth, a new set of hormones come into play. One of them is for the production of milk. That's called prolactin. That is for production of milk. And about a day or two after delivery, you find that prolactin is secreted. And then also colostrum. Colostrum is a sticky mucus that comes out of your nipples. All right? And most people don't know that it is good, nutritious, high in IgA substance. They just wash it out in the shower. Okay? But don't do that. Colostrum is good stuff. It's very nutritious, growth factors, low fat, high IgGs. The immunoglobulin A, very important. So at this stage, you've gotten IgGs from mom, and then you now get IgAs from mom as well. Okay? So here you are, you should feed your infant. All right. Now there's another hormone that's produced that's called oxytocin, or the so-called love hormone. In other words, this is also used in the treatment of anxiety, but it's there here for uterine contraction. This and prostaglandins really help in the contraction of the breast for milk, the contraction of the uterus to go back to its original size. So, this is the use of oxytocin. Now then here I want to show you the two methods that I use for genetic testing. Okay, so you are pregnant, you want to know if the fetus is okay. All right. This is one method. This is amniocentesis. In other words, you stick a needle through and then you draw out about 150 mils of amniotic fluid. And in the amniotic fluid, there are cells, there are live ones, there are dead ones, and then you do your test. Right? This is the conventional, inexpensive method. However, this is not good when there is a problem, because by the time you have your test results, that will be about 4 months, all right? And it's not like, in order to deliberate and to consider alternatives, you can't just do it overnight. You have to talk to your friends, your family, your priest, lots of things. So while this method works well, it's sometimes not good enough. So there's another one. This is called chorionic villus biopsy, and you can take these cells not from the amniotic fluid, but from the placenta. All right. This is much faster. You can do it in 8 weeks, and you can do the same test. It's a little bit more expensive, and it requires a little bit more expertise because you're drawing cells from the placenta. Now then there is this thing, I added this here. This is something I told you about in the past, and that is if mom is Rh negative and the fetus is Rh positive, that means mom is making antibodies against the fetus. All right? This is called erythroblastosis fetalis. And you can review this because I've talked about this before. Now then my last word to you, as far as this is concerned. At this stage, after the birth of your child, all right, within 1 or 2 days, your breast is huge. All right? It's turgid. It is the most beautiful it will ever be. Okay? So therefore, bring your cell phone along. Okay, that's it. So, all right, so I'm stopping here. Okay, let me say a few things. I hope you see, filled out the OCQ. Remember, 70%. I'll give you an extra one.